All right, let's wrap up lecture 8 with lecture 5, action potentials. So action potentials are for fast communication that have to travel long distances. For example, if I have a neuromuscular junction, right, that means that I have now a neuron, its axon and its axon terminal is going to innervate muscle. And this is how I can now have movement in some place of the body. Now, we also have communication to glands. So remember in unit 1, we were talking about that anticipatory insulin release. And that allows us now to pre-secrete insulin, even though we don't have any glucose stimuli in the body, in order to anticipate for when you're going to have your next meal. And so those signals will come from the brain, but I have to have an action potential, travel down a neuron, and then get that neurotransmitter to be now secreted and then bind to its cell type in the pancreas in order to secrete that insulin. And then, of course, the classic example is the heartbeat, right? If we want muscle to contract, it first has to receive an electronic stimuli. Okay? It needs electricity. Electricity first, and then muscle contraction will follow, right? And so here are your main examples of what goes on and what is the output right after we have an action potential. Before I go into the mechanism of action potentials in the axon, I first want to make sure that we are seamless in understanding that graded potentials will initiate an action potential. Right? So here is our first picture, and what you folks are seeing here is the voltmeter measuring right at the point of stimulus, a little bit farther away, like the mid cell, and then right before the axon hillock, which is also called the trigger zone. Okay? Now look at the measurement, right? So look at the stimulus. I receive stimulus, which is a neurotransmitter binding to its ligand-gated channel, letting some type of ion in. And in this case, we can see it's a positively charged ion, right? It's because I'm having depolarization of my cell membrane. Now remember, graded potentials can be in the dendrites or in the cell body, okay? Now look, I just finished telling you folks in the previous lecturette is that these ions are going to travel down their concentration gradient, and they're not going to just travel in a straight line, people, right? They're going to go all over the place. They're going to travel. They're going to go here, there. They're going to bounce everywhere, right? So it makes sense that as I get farther away from point of entry, that signal is going to start to die out. So look at this graph, right? What is 55? Negative 55 in this dot, this is telling you what the threshold is, okay? And so if you look mid-cell, did we start to lose too much charge? We did, okay? And by the time we now get to the axon hillock, aka the trigger zone, are we going to have an action potential? No, because we didn't hit that threshold voltage that is needed to open up all the sodium voltage gated channel that resides in this area, okay? Now, look at this example. In this example, we have a lot of stimulus. So think about what that means physiologically, okay? Go back to your mantra. Ligand plus receptor equals cellular response, okay? If I'm going to have a stronger stimulus, what do I need? I need a lot of ligand-gated channels, and I need more neurotransmitters, okay? Those two dance together. You need both. So if I have more neurotransmitters binding to more ligand-gated channels, I can get even a larger stimulus. More positively charged ions can come into the cell. And of course, they're going to do their thing as all small molecules do. Travel down their concentration gradient. But if you get enough, a big enough amount to start, then by the time you've now lost charge, because remember, charge likes to move everywhere, and if you get high enough charge at the trigger zone, aka the axon hillock, I can still maintain enough charge to hit that threshold, and then I'm going to have now the initiation of that action potential, okay? We've now melded how graded potentials initiate action potentials. And I've shown you action potentials only occur in the axon, okay? Because now, once I've now, what we say integrated, think about what that word means too. Integration means make a decision, okay? Remember, this cell, this neuron is receiving negative signals, positive signals. So I need to have the summation of all those signals at the integration area, aka axon hillock, aka trigger zone, in order for that neuron to decide, do I pass on the signal or not? Okay? And if I do pass on the signal, that means that all the signals I received from other neurons are strong enough for me to hit that threshold. Okay? Let's move on. All right, let's talk action potentials, people. Okay? So what this top picture is showing you folks is an analogy. If this works for you, you run with it. If this doesn't work for you, you close your ears. Okay? Now, the analogy of an action potential is the following. It's much like when you stack dominoes, and when you have one domino fall, it's going to hit the second domino, which causes the third domino to fall, so on and so forth. So you get this tat, 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 which allows you now to send a signal or pass on a signal, if you will, all the way down the axon. Okay? Now, let's go to the axon. So look at what you folks are seeing here, right? There are many voltmeters, 1, 2 to 11, okay? So right now, what this is showing you in time, what these voltmeters are going to be reading, okay? AKA electrodes. Electrodes is the needle portion that you stick into whatever you want to measure the volts. Like I showed you folks with the cucumber. All right, now look at the electrode number 1. What you folks are seeing is an action potential, okay? Is what you're seeing now. Look at the membrane potential. Okay, this is important, right? We got to be able to read these graphs. Now, the membrane potential is becoming depolarized, right? Is because it's becoming more positive, right? You folks see this? Yes? Let me just write here down. I was going to write this way, but I got dizzy. Okay, more positive, okay? As you go up on the arrow. Depolarize. All right, now look at electrode 1. I'm measuring the voltage right here in this portion of my axon, okay? Now remember, the trigger zone, if I reach threshold about negative 55, negative 50, it varies, but around negative 55, let's just say, then I have a whole bunch of sodium voltage gated channels that are going to open. Okay, which means that I'm going to have sodium rush through these channels, and then I'm going to have now sodium start to rush into the axon, because remember, small molecules will diffuse from high concentration to low concentration. And so as sodium now is entering, what's going to happen is you're going to see depolarization, this positive charge. Okay, now as area number 1 becomes more positive because of sodium, right? What's going to happen at number 2 is I'm getting an influx of sodium, or a positive charge in this area. And the reason being is in each of these nodes, right, is that you have more sodium voltage gated channels. So as soon as you have a positive charge that reaches about negative 55 volts in this area, you're going to open up these gates, and guess what? Guess who is going to come rushing in? Sodium. And this is happening at each and every node. So what you folks are seeing here is at areas 1, 2, 3, and 4, sodium has come in, opened up those voltage gated channels, yes? And sodium now is rushing in to get that depolarization. Now, what you folks are seeing on this bottom side, right, is that we're starting to lose that positive charge. And why is that? You already know. It's because on this side is that I have sodium potassium pumps that are constantly pumping the sodium back out so I can do this process again, okay? Now, when you get to number 11, this dotted line, we haven't had depolarization yet. This is the potential of what can occur. We haven't gotten there yet because I don't have enough positive charge to open all of the sodium voltage gated channels that are in this node, right? And this node is the node of Ranvier. Okay, that's from anatomy. All right, let's expand on this. One thing before we leave this slide. Look at all of these little action potentials. You see them? What can you folks see? I'm kind of giving it away. If I could draw a straight line, they are all the same amplitude. Amplitude means height. And they're same amplitude, and they have to be the same amplitude. Think about this graphically, okay? In order for me to have the same amplitude throughout, that means that I have to have I have to have about the same amount of sodium gated channels and the same amount of concentration gradient that's allowing the same amount of, relatively the same amount of sodium coming in. And the anatomy tells you the function, right? If I have X amount of sodium gated, voltage gated channels, I have a specific gradient, then I'm going to hit this same amplitude all the way down the axon, okay? Now, an action potential is what we also call all or nothing. Meaning that you're either going to have an action potential or you're not, okay? And action potentials will start at the axon hillock, because once I reach threshold, I will let in enough sodium to start kicking off all of these voltage gated sodium channels like dominoes, boop, 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 until I reach the axon terminal, right? So this is a axon terminal, and at the axon terminal, all of this sodium will eventually lead me to have the release of more neurotransmitters. All right? Okay, let's move on. All right, let's talk about the resting potential of the axon. This is before an action potential has occurred. And sorry, AP stands for action potential. Okay, now here's our cell again. All right, and so in our cell, we're going to have sodium concentrations outside in very, very, very high amounts, but sodium small amounts inside of the cell. Potassium is going to be in huge amounts inside of the cell, and then small in the outside of the cell. Now, how are these concentrations maintained? Go back to your basics. You folks already know. ATP pump, right? And so the ATP pump, specifically the sodium potassium ATP pump, is 3 sodiums out, 2 potassiums in, okay? And that gives you that gradient. This pump don't stop until you die, okay? So just as long as you're making ATP in your body, and you are because you're listening to this lecture, is that your cells are constantly pumping sodium out, pumping potassium in, pumping sodium out, pumping potassium in. Okay, so remember that. So at rest, resting potential of the axon is going to have these concentration patterns. Now, let's go to the membrane potential. Remember, we're talking about action potential. So this is all happening where? In the axon, right? Now let's draw some lines. So here, this is -70. So you folks know this is the resting potential. And then about -50, -55, and I don't want to confuse you folks, but just threshold -50, -55, right? So it's different in different textbooks. But that's threshold. What does threshold mean again? Threshold is the voltage needed to open up a voltage gate. Okay? Now, let me show you folks this line. This is an action potential. This is happening, and let me just draw it out for you folks. Let me draw it here because I have to make some notes. So remember, this is the axon, yes? And I have one electrode right here. And I'm measuring the voltage inside this specific portion of the axon. Okay, just this one. That's it. That's why you see one line. Now let's talk about the mechanism of how we get this change in voltage. So we're going to kick things off with this portion of the line right here, right? This little flat line at -70. You folks know that now the action potential has not started yet. Our axon is at rest, okay? That's the resting potential. But if we now integrate, meaning that if the graded potentials that are received from a single neuron are strong enough when they finally get to the axon hillock and they open up all of those sodium gated channels, we will get a signal that will definitely surpass threshold. Okay. And so here I have now depolarization happening, right? And how does depolarization happen is we hit threshold, we're going to open up sodium voltage gated channels. Okay. That's my notation here. I wanted to write out sodium voltage gated channels, but then my graph gets too crowded and you can't really see. So please make sure that you understand V channels. It's really voltage gated channels. Okay. I hit threshold. I have a voltage that is positive enough to open up these voltage gated sodium channels. And of course, because sodium is constantly being pumped out by the sodium potassium pump, I have a gradient. And then that will allow me now to have sodium come rushing into the axon. Okay. That's what's happening here. Now look at this graph. Okay. Now, if this is depolarization, I'm going to depolarize until I hit a limit, right? Because what happens here is that at this voltage, it's very positive here. Yes, positive 30. Those channels are going to close, right? And so when they close now, sodium is not going to be able to come rushing into the cell, or the axon, I should say. And so here I'm going to stop at this particular voltage. Now, at around positive 30 millivolts, guess what? That is the perfect voltage to open up potassium voltage gated channels. And they open up. And guess what? Don't forget the slide before. Memorize that because that's the concentration gradients that are set at resting. Okay. Now, because potassium is very high inside of the cell and low outside of the cell, what's going to happen here is potassium is going to diffuse out, traveling down its concentration gradient. This is called repolarization. It's because I'm repolarizing. I'm coming back to becoming more negative. Okay. So take note of the notation. In graded potentials, I had depolarization, hyperpolarization. In action potentials, I have this depolarization and repolarization. Okay. Now, what's going to happen right here is I'm going to have now the voltage to where the potassium voltage gated channels are going to close. Okay. They close here, right? And so what's going to happen is I start to close my voltage, potassium voltage gated channels, but I have this overshoot. Okay. And this overshoot is something that we call hyperpolarization. Okay. And we get overshoot is because the potassium channels, they close pretty slow. Right? And so we get some extra leakage of potassium that's running out of there. And that causes this dip way below our actual resting potential. Okay. Now the hyperpolarization area is very important because it helps us propagate the signal forward. Right? So if I am now, if this is my axon here, and then if this is my cell body, right, I can only have an action potential that's going to travel down towards the axon terminal. Okay, I can't have it go the opposite way. It's because of this area of hyperpolarization. Now, this area of hyperpolarization is called the refractory period. Okay, and so this is an important part of the action potential. It's because it does not allow us now to have a signal going backwards towards the cell body. And let's talk about specifically what's going on during the refractory period. All right, let's talk refractory period, right? And so here, remember, the refractory period is defined by the amount it time it takes for an excitable membrane to be ready for the second stimulus. Okay, so let's write that down. So we're going to say time it takes to be able to have another action potential. I reworded it into my own words because that other words I said was a textbook sentence and I don't understand it as well as my own. Okay, so here we go. So the refractory period is the time it takes to be able to have another action potential. It's because during the refractory period, a couple things are happening here. Okay, so one of the things that is happening is I have to reset all of the sodium and potassium voltage gated channels. People, I'm so sorry. I don't really like acronyms too much, but I'm running out of runway. So can you put voltage gated channels, or if you put VGC, just know what that is. Okay. So I have to reset these sodium, potassium voltage gated channels. And what that means is and I don't want you folks to memorize the mechanism of resetting the channel, but I am going to draw it out for you folks. And forevermore, we're just going to call it reset the channels, right? So if you're a voltage gated channel, right? So if this is you, boop, 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 you have this like ball and chain kind of a thing. And the ball should be blocking the hole, but I'm not going to block the hole because I want you folks to see what I've drawn here with my computer pen. Now, make pretend that this is closed, right? So if I want to open it up, I've now swung that part of the protein out, and then now I can have sodium or potassium or whoever is coming in and out of this voltage gated channel. You folks with me? Yes? Now, I need to reset all these channels, meaning that they have to come back to position 1, right? Now, in order to get back to position 1, the channel has to go through all of these different conformations, right? So it has to do this weird kind of swing down, and then it can swing this way, so on and so forth. Just know it has to do all these weird little movements first before it can get back to being reset and ready to go. Okay, back to position 1. That means, this is what I mean by resetting the channel, okay, is that it has to undergo multiple protein conformations in order to come back to rest, okay? And it takes time, right? This takes time in order to do so. Another thing is I gotta go redo or remake all the concentration gradients. Yes? And this is all via the sodium potassium pump. Because think about it. Look at this line. What is this line? This line is depolarization, which is via mechanism dictated by me opening up sodium voltage gated channels, okay? And in order for these voltage gated channels to be receptive to voltage, they need to be in this first conformation, okay? The second thing is, in order for sodium to move, to depolarize the axon, I have to have a concentration gradient. So in order for this line labeled A to occur, I have to have all my channels reset to conformation 1. And I have to have a concentration gradient. Because think about it. What has happened in A? I let all of my sodium, where? It went all the way inside. It went all the way inside the cell, all of it. So in order for me to depolarize again, guess what I got to do? I got to get all that sodium back out. And so that's what's going on in regards to this refractory period. Okay, are you folks with me? Now, I want to show you folks here in this area here is the refractory period can be broken up into 2 portions, right? So I'm just going to draw this line in the sand, if you will. Now, absolute refractory is this is the period of time where you can absolutely cannot have an action potential. And think about why. Think about why that is. And if you don't know, go back to the mechanics, go back to your foundations, okay? Because everything that you need to know to answer this question of why can't you have an action potential during this part of the refractory period is all right here, okay? I'm gonna let you folks think about it, okay? Now, in the relative refractory period, look at what has happened. A little bit of time has gone by, right? What's happening to my charge? I'm starting to do what? Come back to resting, right? Now, in the relative refractory period, I can have an action potential. But I really need a strong signal from the axon hillock to propagate a signal in this area for relative refractory period. And why is that? Go back to your foundations again, right? You should be asking yourself, what do I need to have in order to have an action potential? I need to have all of my sodium voltage gated channels set at conformation 1, and I need to have a sodium concentration gradient, okay? Think about what's happening here in the relative refractory. I'm going to have you folks think about it. So much so that in our practice problems, I'm going to ask you folks, what is the amplitude of an action potential that is fired during the relative refractory period? Is it smaller, larger, or normal? And I want you folks to explain using the information that I gave you, because it all boils down to mechanics, your foundations. Everything you folks know foundational is what you need to answer all these problem-solving questions. People, you did it. That's lecture 8. I'll see you back for lecture 9.